Thank you, Asif, for the wonderful presentation. I also would like to thank SBIA staff for organizing the event. Hello, everyone. Today, I'm going to speak to you about in vitro drug release testing for complex generics from a bioequivalence perspective. Here is the outline of my presentation. I will discuss the golden circle for in vitro drug release testing. The first question: why do we conduct IVRT? What kind of role can IVRT play during product development and approval? The slide here lists 4 key objectives of IVRT, including assessing the effect of formulation factors and manufacturing methods on drug product, routine assessment of quality control to support batch release, establishing an in vitro in vivo correlation or relationship, IVIVC, IVIVR, and supporting evaluation on bioequivalence. I will be focusing on IVRT for BE purpose in today's talk. When IVRT is used to support bioequivalence assessment, it is considered as a performance test evaluating sameness in the rate and extent of drug release between test and reference products. So far, IVRT has not been used as a standalone study. Rather, is used as one component of a totality of evidence approach. IVRT has been recommended as part of an in vitro approach to detect variations in formulation and related physical-chemical characteristics. It has also been recommended in conjunction with in vivo BE studies to capture potential differences in formulation that may not be captured by the in vivo studies. This slide summarizes when IVRT is used as part of an in vitro approach for establishing BE. In terms of dosage form, in vitro approach has been recommended for ointments, emulsions, and suspensions for various routes of administration, including ophthalmic, topical, and parenteral. This slide shows the PSG on risperidone PLJ-based microsphere product, and this is the only PSG where the IVRT is recommended in combination with an in vivo study. The purpose of including IVRT in addition to the in vivo study is mainly to evaluate the initial burst release phase as well as the lag phase. Next, I'm going to switch gear to discuss how to develop and validate an IVRT method. This can be further divided into 3 questions. First, what are available IVRT methods? Second, what are the parameters to consider for method development? And third, how should the IVRT method be validated? This slide shows nearly all available IVRT methods for various dosage forms. I'm not going to discuss each one of them. The point is that there are many different methods available, and in order to select the most suitable method, we really need to understand the dosage form and what are the potential challenges for developing and validating an IVRT method. Different physical form of the formulation requires different features. For instance, USB apparatus 1 and 2 are developed for conducting IVRT of tablets and capsules. They are not readily applicable for conducting IVRT of liposomes or emulsions, as those systems need to separate the released drug from the formulation. Besides the physical form, there are also other constraints associated with IVRT, such as the desired release rate and drug solubility and stability. All those things need to be taken into consideration when developing an IVRT method. This slide presents some of the common experimental parameters for developing IVRT methods. By no means this list is exhaustive. The key is that you should always provide sufficient justification in your development report for the selected experimental parameters. When the developed IVRT method is fully validated, it should be discriminatory and reproducible. The reproducibility means the method is precise and robust. The discriminatory ability includes sensitivity and selectivity. A fully validated method should be able to capture differences in drug release resulted from differences in some physicochemical properties of both the drug substance and the formulation, such as distinguishing different polymeric form, excipient grade, source. In addition, the method should also be able to detect difference in formulation attributes affected by manufacturing. I will further elaborate on this in later slides using 3 case studies. The first case study is about IVRT of suspensions. It is well accepted that particle size and size distribution as well as viscosity of the formulation are key formulation characteristics that affect drug dissolution rates. To demonstrate discriminatory ability of an IVRT method, you should intentionally introduce meaningful variation into either the formulation composition or manufacturing process of your test product. In the current case, in-house formulations with varying particle size distributions relative to the reference product were prepared to validate the IVRT method to be discriminatory in terms of particle size distribution. Aforementioned, there are generally more than one options when selecting the dissolution apparatus or settings. When using the USP2 apparatus, the release rate was too fast. All formulations showed nearly identical release kinetics. When conducting the IVRT based on vertical diffusion cell, which involves use of a membrane, it was observed that the release slowed down due to the presence of the membrane presenting a physical barrier to the drug diffusion. However, discriminatory ability is still lacking. So, the challenge is that how to improve discriminatory ability of a method. In most cases, IVRT is conducted under a sink condition. However, in certain cases, in order to improve discriminatory ability and or make experimental setting more biorelevant, IVRT under non-sink condition may provide advantages. There may be concerns on whether IVRT conducted under a non-sink condition would be considered acceptable. The answer is sink or non-sink condition. Either way may be acceptable as long as sufficient justification is provided. In addition to considering a non-sink condition, having capability of continuous monitoring during IVRT can also be helpful for improving the sensitivity selectivity of the method. Figures here show the discriminatory ability of the IVRT method developed by my colleagues in the Office of Testing and Research based on a non-sink condition in combination with continuous monitoring. The nanosuspensions completed release within 6 seconds, whereas the micron-sized suspensions dissolved in 2 minutes. A good correlation between drug release rate and particles distribution was observed. If you are interested in knowing more details of the study and the method, please refer to the reference included at the bottom of the slide. The second case study I would like to briefly discuss with you is IVRT of semi-solids. There are 3 setups recommended by the USP for semi-solids, including vertical diffusion cell, immersion cell, and USP4 with semi-solid cell. With regard to which one is the most suitable setup, one needs to know that there are different types of semi-solid in terms of the properties of the base and the related drug release mechanisms. Unlike other dosage forms, drug release from ointments is determined as the amount of drug release rather than cumulative percentage. Generally, the release kinetics is recommended to fit Hikuji release model and using T/R ratio to compare reference and test products. Due to the time constraint, I'm not able to dive into more details about validation of IVRT for semi-solids. I would like to point out a few common issues associated with method development and validation, such as membrane inertness, receptor median solubility, sensitivity, and robustness of the method. My colleague Dr. Sam Rainey and his collaborators have published a nice research paper describing a comprehensive approach to qualify and validate the essential parameters of an IVRT method for a cyclovir cream, which is a good reference when developing and validating IVRT methods for semi-solids. The last case study I'm going to discuss is IVRT of emulsions. Here is a nice illustration of emulsion formulation design developed by Dr. Xiaoming Xu from the FDA. As shown, emulsions contain multiple components which likely lead to multiphasic drug release. In other words, IVRT of emulsions is complicated. What are the key things to consider when developing an IVRT method for emulsion? First, formulation composition. How much oil and surfactant are used in the formulation? Second, what is the particle size distribution of the oil droplets? Third, very likely, the method needs the ability to separate the released drug from the formulation. And fourth, mass balance to ensure the observed drug release is truly from the released drug, rather than sampling the oil droplets. This last point is often overlooked. To do so, you could monitor both the amount of drug released and the amount of oil diffused through the membrane. This slide describes a couple of common issues we observed when reviewing IVRT of ophthalmic emulsions. First, IVRT method development and validation report is often incomplete. For instance, justification for experimental conditions is lacking, although certain things may appear to be obvious, but from a regulatory standpoint, the scientific assessment can only be done based on provided data. So it is very important to make sure sufficient justification for the experimental conditions is included in your submission. Second, the release data is plotted as the amount of drug released per unit area against square root of time, as vertical diffusion cell is used for conducting the IVRT. However, IVRT using vertical diffusion cell does not necessarily mean the data should be plotted as such. In general, we recommend report IVRT data on emulsions based on cumulative release versus time. Based on the observed common issues associated with IVRT of complex drugs, it is recognized that compendial IVRT methods may not be readily applicable to complex dosage forms, as seen in the cases of suspensions, emulsions, and ointments. The low solubility of the drug in release media compared to formulation gives rise to exceptional slow, incomplete drug release. And IVRT components such as membrane can be the rate-limiting step, reducing discriminatory ability of the method. In the next couple of slides, I will try to summarize some of the key considerations when developing and validating IVRT methods. First, is it acceptable to develop an IVRT method based on announcing condition? The answer is yes, as long as the method is properly validated. Second, which formulation should be used for IVRT validation? In general, in-house developed formulation with meaningful variations in formulation and manufacturing parameters should be used for method validation. Key characteristics of the target test product and the reference product are used for selecting meaningful variables. When selecting critical quality attributes for method validation to demonstrate the method is discriminatory, one may wonder whether it's necessary to investigate all critical quality attributes. My personal view is that it may not be possible to develop an IVRT that can discriminate all CQAs. That being said, I think all CQAs should be considered, and justification should be provided to support inclusion-exclusion of certain CQAs. When a CQA cannot be evaluated by IVRT, then the applicant is encouraged to develop a separate characterization testing to evaluate that CQA. Here are some general expectations on IVRT. When an IVRT method is used to support bioequivalence assessment, it is not intended to mimic the in vivo administration environment or predict the therapeutic effect of the drug. Although an in vitro-in vivo correlation is always desired and is often recommended or encouraged, it is not required. The IVRT should be able to discriminate batches that are not bioequivalent. In general, drug release profiles should be complete, reach a plateau, which means no significant increase over 3 consecutive time points, or achieve at least 85% release. If incomplete drug release is observed, additional information to explain the reasons for incomplete release should be provided. With regard to analytical methods for determining drug concentration in release samples, the ICH or FDA bioanalytical guidance should be followed. Last, I would like to share a few take-home messages with you. Selection of an IVRT method depends on the physical form of the product. Complexity in IVRT generally is due to need for separation and other constraints. To design a good IVRT method, it starts with understanding the impact of formulation and manufacturing process parameters on drug release. Formulations with intentional and meaningful variations are good testing samples to verify that the IVRT method is discriminatory and reproducible. Understanding the mechanism of release and factors controlling release can guide the development and improvement of the IVRT method. Last but not least, I would like to thank my team members, my division and office managements, and my colleagues from Office of Bioequivalence and Office of Testing and Research for their input, help while developing today's talk. Here comes the fun part. I would be more than happy to address any questions you may have, but first let me ask you a couple of challenge questions. First one. Which of the following statements are true? A. IVIVC is required to justify the use of IVRT for supporting bioequivalence. B. All critical quality attributes should be considered when validating an IVRT method. C. IVRT should be able to discriminate batches that are not bioequivalent. D. IVRT should be validated against the reference product. I'll give you all a minute to select your answer. Great job, everyone. I can see you all have selected the correct answers, which are B and C. All critical quality attributes should be considered when validating an IVRT method, although not all are necessary to be included. C. IVRT should be able to discriminate batches that are not bioequivalent. Challenge question number 2. It is acceptable to develop an IVRT method based on a non-sync condition as long as the method is properly validated. Is this statement true or false. I'll give you a minute to make your decision. Wow, you guys are amazing. Everyone got the correct answer. It is true. It is acceptable to develop an IVRT method based on a non-syn condition as long as the method is properly validated. I'm super impressed by how well everyone did with my challenge questions. It will be my turn to answer any questions you may have, and thank you very much for your attention. Next, I would like to pass the podium to Dr. Hiram Patel for his talk.